So we will now discuss the DNA structure. As we learned before from the Watson and Crick DNA model, we know that DNA consists of two complementary anti-parallel nucleotide strands. These two strands form double helix, alpha helix. Now we will discuss primary, secondary, and tertiary structure of DNA. Primary structure refers to nucleotide structure. Secondary structure refers to the helical configuration, three-dimensional structure, and tertiary structure is outlined in the chapter 11 and refers to the complex packaging arrangements of double-stranded DNA within the chromosomes. So let's now talk about primary structure of DNA. DNA is a polymer, which means that it's made up of repeating monomers. Monomers are the nucleotides. So DNA is a string of nucleotides which are joined together by phosphodiester linkage. Phosphodiester linkage is formed between sugar and phosphorus and it is covalent bond. So nucleotides, the monomer, the building unit of DNA, is composed of sugar, nitrogen containing base, and a phosphate. So first we will talk about a sugar. Nucleotides of both DNA and RNA contain sugars called pentose. Pentose sugars have 5 carbon atoms which are numbered from 1' to 5', the same for both molecules. Now, difference between DNA and RNA is that DNA contains deoxyribose and RNA contains ribose. Deoxyribose has hydrogen attached to 2' carbon, while ribose has hydroxyl group attached to 2' carbon. This seemingly minor chemical difference is actually recognized by most of the cellular enzyme which interacts with DNA or RNA, such as DNA polymerases versus RNA polymerases, giving the specific function to each of these nucleic acids. In addition, this one extra oxygen atom in RNA molecule makes it more reactive and less chemically stable than DNA, and therefore DNA is better suited to serve as a long-term storage of genetic material, serving as our blueprint. The second component of nucleotide is its nitrogenous base. There are two types of the bases, purines and pyrimidines. Each purine consists of 6-member ring attached to a 5-member ring, while pyrimidines have only 6-member ring. Now, purines are adenine and guanine, and that you find them in both in DNA and RNA. Pyrimidines are cytosine, thymine, and uracil. Cytosine is common in DNA and RNA, while thymine is found in DNA and uracil is found in RNA. If you compare members of each group, whether you compare 2 purines or 3 pyrimidines, but you have to realize that they have common structure. What they differ at are the functional group attached to certain carbons and double bonds, position of double bonds. So those are the differences between these 3 pyrimidines, and the same apply for 2 purines. A nitrogenous base always forms a covalent bond with the 1' carbon atom of the sugar in the nucleotide. If you take a deoxyribose or a ribose sugar and put it together with a base, Together they form nucleoside. The third component of the nucleotides is phosphate, which is outlined right here. Phosphate is composed or consists of a phosphorus, which is bounded to 4 oxygen atoms. Phosphate groups are found in every single nucleotide, and as you can see, very frequently they can carry negative charges, which makes DNA negatively charged. This is very important because this negative charge of DNA enables DNA molecule to interact with positively charged histones, and we will talk about that later on in following chapter. Now, all three components, the sugar, phosphate, and base, form deoxyribonucleotides or ribonucleotides in RNA. The proper name would be in DNA deoxyribonucleoside 5' monophosphate. So, if you look in the composition, phosphate is always attached to 5' carbon, and base is always attached to 1' carbon of the sugar, of the pentose. Now, since we have 4 different bases, there are 4 different deoxyribonucleotides: deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate, and deoxycytidine monophosphate. This table just summarizes the names of DNA bases, nucleotides, and nucleosides. I will not go over it. You can very easily read it. One thing I would like to point at is difference between nucleotide and nucleoside. Nucleotide is in DNA, let's say nucleotide which contain adenine is deoxyadenosine monophosphate. That's a nucleotide. Nucleotide is composed of sugar, phosphate, and nitrogenous base. Nucleoside is deoxyadenosine. Nucleoside is composed of sugar and nitrogenous base. Okay, so when we will talk about, let's say, substrate for DNA synthesis, the substrate is deoxyadenosine triphosphate, which is different from nucleotide, which is deoxyadenosine monophosphate. So keep it in mind that apply to all of them, to guanine, thymine, and cytosine. So now we know a composition or structure of nucleotides. So let's review again the primary structure of DNA. So primary structure of DNA refers to the sequence of nucleotide connected by covalent bonds and forming side chains of DNA and chain of RNA as well. The nucleotides are connected together by so-called phosphodiester linkage, which is covalent bond. This covalent bond is formed between 5' phosphate with 3' hydroxyl group. So new nucleotide can be added to the DNA strand only. It can be added and attached to 3' hydroxyl group via 5' phosphate. This orientation gives a polarity of DNA molecules. It has 5' end and 3' end. 5' end has a phosphate attached to 5' carbon of the terminal sugar, and 3' end has hydroxyl group at the 3' carbon of the sugar. The same polarity you find in RNA molecule, where you have at 5' end the phosphate and on 3' end you have hydroxyl group. In DNA, you can see that 2 anti-parallel strands are connected via hydrogen bonds formed between nitrogenous bases. And you can see the differences between adenine-thymine bonds and cytosine-guanine bonding. Adenine and thymine, right here, are connected via two hydrogen bonds. Cytosine and guanine forms three hydrogen bonds. What does it tell you? The DNA molecule which has more cytosine guanine pairs would be more stable than DNA molecule which has higher percentage of adenine and thymine. What I mean stable, you know that you can denature DNA breaking down hydrogen bonding. The hydrogen bonds between two strands of DNA are functional. Hydrogen bonds is not strong bonding. However, DNA molecule is a stable molecule because you have many of these weak bonds. However, it would not be smart from evolution to introduce covalent bond between two strands. Why? Because DNA needs to replicate. DNA is transcribed. So DNA needs to unwind. These bonds between two strands has to be broken at certain point. If it was covalent bonding, it would cost cell too much energy to break bonds between two strands. So the hydrogen bonding is much more efficient organization of the or connecting of two strands together, while as you recall the strands, the primary structure connection of nucleotide which we need to have as a stable bond are covalent bonds. So if you have DNA and you need to denature it, you want to break apart two strands, you will need higher temperature, more energy to break it apart if it contains more cytosine guanine pairs than if the DNA molecule contains more thymine adenine pairs.